Hello and welcome to this Revision Monkey video on what's going to be in the Combined Biology Paper 2 exam for 2023. This video is for pupils studying the AQA specification and those who are studying the Foundation Tier paper. Let's have a look at what to expect in Biology Paper 2. So again, we'll very quickly recap compare, explain and evaluate questions. We'll look at a little bit on graphs, a little bit on tables because you need to expect lots of tables and graphs in your biology paper. We'll have a look at some of the calculations to expect. About 10% of the paper will be calculations for biology paper 2, and also 15% of the exam will be about required practicals, so we'll have a look over those ones as well. So as a quick reminder, for evaluate questions, they won't tell you this, but you must include advantages, disadvantages, and a justified conclusion in your answer. They will just have the word evaluate in the question, but it means give advantages, disadvantages, and a justified conclusion. Compare questions, again, they won't tell you, but when you answer compare questions, you need to write similarities as well as differences in your answer. And explain questions, you must say why something is happening, so include as much scientific detail as possible. So I do go into a bit more detail in these in the first biology video, so feel free to look back there if you want to look at these evaluate, compare and explain in more detail. So in biology paper 1 we did a little bit of work on graphs. Just to add to that, you need to read the graphs really carefully. So if the question asks what is the rate of photosynthesis when the light intensity is 400 lumens, they'd expect you to show you're working on the graph by drawing a line up from 400. But my point is, if when you draw the line up, you meet the line on the graph and it is not exactly on the corner of a square, that is okay. Just continue by drawing a line across, and you can see this one, it doesn't come off at 30, it doesn't come off at 31, so we need to be really accurate that our answer is going to be 30.5. Don't force it to meet one of the lines on the graph. If you find that it comes in the middle of a box, that's absolutely fine. Just read it accurately rather than forcing it up here to the next line. Also, I just want to look at describing graphs. So this one says describe the relationship between light intensity and the rate of photosynthesis. So you're looking at 3 different shapes on the graph. First bit is the steep bit at the start. So you describe that by saying between 0 and 300 lumens as light intensity increases, the rate of photosynthesis increases rapidly. So there's the key word there, underlined, rapidly. And then you have to look at the next part of the graph. And between 300 and 560 lumens, as light intensity increases, the rate of photosynthesis increases gradually. So this is a more shallow line, so we call it a gradual increase. And then finally, looking at the last part of the line, after 560 lumens, as light intensity increases, the rate of photosynthesis stays constant. So a really, really common question for biology paper 2, but it could come up in chemistry or physics as well. There's more information on graphs in biology paper 1, but I'll assume you've had a look through that already. Again, I did a lot of examples of tables in biology paper 1 video, but I just wanted to add that they might give you a blank table to fill in. So they give you all the information in the question and ask you to complete a blank table. So this question says a student carried out a transect. They placed a quadrat every 50 centimetres from a tree, and they counted the number of daisies in each quadrat, and their results are below, and they'll give you a list of results. And then it says complete the table to show the student's results. One result has been completed for you. Sometimes they'll complete the first result to give you a clue to put your headings in the table, and sometimes they don't, so look out for that. If they don't, you must make sure you've got the headings in. So you'd have to complete the table by writing in appropriate headings. So it said they placed the quadrat different distances from the trunk, so let's call this distance from the trunk in centimetres, and they counted the number of daisies, so you put that in the right hand column, and you're going up in 50s as the question tells you, And in this column, you're just writing in the order that they've given you the results as they have collected them. But again, there's more on tables in paper 1. If you didn't get to watch that video, have a look back and watch those first few minutes. 10% of biology paper 2 is going to be calculations. So this is a calculation which is linked to a required practical, so I just wanted to go over this one. It's about estimating population size. So the question says, estimate the population size of snails in the 30 metres by 25 metre woodland. That's represented over here. And the student used a 0.25 metres squared quadrat, and there were a mean of 3 snails per quadrat. So just bear in mind, the quadrat, if it's got metres squared, that's already the area. You don't have to do anything with that, it's already the area. If they give you the quadrat as a diagram like this, you might have to work out the area by multiplying 0.5 by 0.5, as would be this example. So if it's got metres squared, the area has already been calculated for you. If they give you a diagram to show you the quadrat, you would need to calculate the area first by multiplying the length of both sides. So reminder of the equation that we need to use is population size is equal to field area divided by quadrat area multiplied by the mean number of organisms per quadrat. Because this first bit will tell you the number of quadrats that would fit in a field. So the field area divided by the quadrat area would tell you how many of these quadrats would fit side by side in the field. And then if there was a mean number of 3 snails per quadrat, multiply that by 3, and that will give you an estimation of the population size. So if we have a look and calculate the field area, to give you 750 metres squared, we've got the quadrat area already, number of organisms are 3, so putting our numbers in, field area 750 divided by 0.25 multiplied by 3, showing our workings out and ending up with our population size of 9,000 in this example. So look out for that one if that one comes up in the paper 2 exam. Also, uncertainty can come up on any paper. Uncertainty is calculated by the range divided by 2. For example, this is a set of results with 2 people, A and B, and it measured the distance the ruler fell through their hand. So they've taken 3 repeats and calculated the mean. You'll notice the mean is the same for both people, but the uncertainty will be different. So uncertainty in the person A's case would be 7 minus 5 divided by 2, because the biggest number is 7, smallest number is 5. That gives us our range, and then divided by 2. So our uncertainty would be plus or minus 1. And then the uncertainty for the second set of data would be plus or minus 4, because the biggest number is 10, smallest number is 2, and that would give us our range, 10 minus 2 divided by 2. So yes, they both have the same mean, but because the person A's numbers were closer together, they have a smaller uncertainty. Because person B's had a bigger range in their results, they have a much larger uncertainty around their mean. So the 2 calculations to be aware of, uncertainty can come up in any paper, and population size calculation will just be relevant for biology paper 2. So the required practicals for combined science are organism distribution, which includes quadrats and transects, and the reaction time required practical.
Reaction time required practical. As reaction time is so quick, it's very difficult to measure with a stopwatch. So instead, we're going to measure reaction time using the ruler drop test. For example, we could ask the question, how does caffeine affect reaction time? Now, this is just one example of a factor that we could investigate. You could well in the exam be asked about something like distractions or tiredness or something like that. So this here could change and become a different factor. So let's go through the method. First of all, you'd work in pairs for this practical. Person A will be the person who is dropping the ruler, and person B will be the person who is catching the ruler. Person B should sit down and rest their dominant hand on the table with their thumb and fingers hanging over the table. Person A should hold the ruler vertically with the base of the ruler in between person B's thumb and forefinger. This is an important control variable because you need to make sure that you drop the ruler from the same height every single time. Person A will drop the ruler without indicating to person B when this will be, and person B should catch the ruler as quickly as possible. If the ruler falls through just a short distance, this would indicate a fast reaction time, and if the ruler fell through a longer distance, that would indicate a slow reaction time. We can then use a conversion table to convert the distance the ruler fell through the hands into a reaction time. So this will turn the centimetres from the ruler into a time in seconds. We would then repeat this 3 times and calculate a mean reaction time. So you've got the reaction time for person B. We're then going to change this factor here, the amount of caffeine they've drunk, and then we're going to repeat the experiment. So the next step, person B should then drink a set volume. It doesn't matter how much you put, but I've just suggested here 200 ml of caffeinated Coke. They should wait 5 minutes. Again, you should just suggest a sensible amount of time here, and then repeat the ruler drop test 3 times and calculate a mean reaction time. Repeat the investigation using 10 other people, so you could first swap around so that person A and B swap around, and then find other people to repeat the investigation.
So if we look at some variables for the investigation, the independent variable would be the volume of the caffeinated drink that was consumed. So in this case, for example, 200 ml or 0 ml were the choices. The dependent variable would be the reaction time, because that's what we're collecting data on. And the control variables, things such as using the same hand when catching the ruler, and also making sure the ruler is dropped from the same height.
Organism distribution required practical. There are 2 different ways that we are going to look at to sample organisms. The first one is random sampling, and the second one is transects. So random sampling might be used to estimate a particular population size. So for example, you might get a question a little bit like this that says, describe a method to estimate the population size of daisies growing in a 200 metre by 100 metre school field. These numbers here will come up a little bit later on in our calculation. So we can't count every single daisy growing in the school field. It would just take too long. So we need to have a method of sampling that is completely random, and if they talk about why in the exam, it is to avoid bias. So as we said, we can't count the number of daisies in the whole 200 metre by 100 metre field, so instead we measure out an area of the field using a tape measure. So here I've got a 5 metre by 5 metre area, for example. We then divide this grid up into squares using string and number those squares 1 to 100. So our grid might look something like this. Then the most important thing with random sampling is we use a grid called a quadrat. Now the one I'm going to use in this example is a 0.5 metre by 0.5 metre quadrat. In the exam it may well be a different size, but that's absolutely fine. And we need to choose where to place our quadrat within this grid. So again, it would take too long to do all 100 squares and count the number of daisies in each square. So what we use is a random number generator to select a square, and you can get those on calculators or phones, and it will pick a number at random for you. So we place a quadrat on that selected square and count the number of daisies present, and then we repeat the steps for perhaps 9 more random squares. So we might do 10 in total. So let's say our random number generator picked us out the number 9. What we do is we take our quadrat and we place that on square 9 and we count the number of daisies present. We would then repeat that for another square. For example, if our random number generator there picked us up 47, we'd place our quadrat on 47, count the number of daisies present, and again, for example, 72, again counting the number of daisies present. Now we'd repeat that for around 10 squares, so we've got different quadrats here, and we've counted the number of daisies present in the squares. And we're going to then calculate a mean number of daisies per quadrat, and because we're going to use this number again in a minute in a calculation, I'm just going to leave it as a decimal rather than rounding it as I would do normally with a mean. So we've got a mean number of daisies per quadrat, and now we need to use this to estimate the number of daisies in the whole school field. So we need to know our area of our quadrat, so we do 0.5 times 0.5, and that's 0.25 metres squared. We need to know the area of the school field, so given in the question it was 200 metres by 100 metres, so 20,000 metres squared. The next thing that you need to work out is the number of quadrats that would fit in the field. So we've got the area of our field divided by the area of one quadrat, and that gives us 80,000 quadrats. So if 80,000 quadrats fitted in our field, and the number of daisies in each quadrat was 0.8 as an average, then the estimated number of daisies in the whole field would be our number per quadrat multiplied by the number of quadrats that would fit in the field. So 0.8 times by 80,000 would give us a total estimate of 64,000 daisies. A couple of key points then. Increasing the number of squares sampled will increase the reliability of the population size estimate. They may use the word reliability or they may use the word accuracy, but the more quadrats that you put down on that field, the more reliable your estimate is going to be. Now, for some organisms, calculating percentage cover might be more appropriate than counting the number of organisms. For example, you may well have seen something like lichen growing on trees or walls. If you wanted to sample lichen, you might use a really small quadrat, so just perhaps a 10 by 10 centimetre grid. In the previous example with daisies, we would look at one random square which we'd selected by our random number generator, and we calculate the number of organisms inside. However, with lichen, we can't do this because you can't see individual organisms. So instead, you might need to count the number of squares covered by the lichen. Now, all of the whole squares you can just count up like so, and then we will look at any partial squares that we've got, like perhaps this one and this one, where they could fit together roughly to make a complete square. And we will continue to do this, estimating the total number of squares that are covered by the lichen. So when we've got this, we use the equation the number of squares covered divided by the total number of squares times 100, and this will give us our percentage cover. So if I've estimated that 30 squares in total are covered by the lichen, there are 100 squares on the grid, so 30 divided by 100 times 100 will give us an answer of 30% coverage. Another way we could sample the distribution of organisms is using a transect. So for this we might be asked questions such as, how does light intensity affect organism distribution? Or, how does the distribution of organisms change as you get further from the shore? So let's take this example in terms of seeing how light intensity affects organism distribution. So under the tree, the organisms will be subject to more shade than further away from the tree. So we can sample the organisms at regular intervals and see how the light intensity affects their distribution. So what we do is we'd lay a transect line, this could be a tape measure or some string, along the ground starting at the base of the tree. We would place a quadrat at the 0 metres mark and count the number of organisms, so for example daisies, or it could be something else that you're looking at, present in the quadrat. We would use a light meter to take a recording of the light intensity and record this in a table. We then repeat steps 2 and 3 at regular intervals along the transect line, for example every 2 metres. So we place our quadrat and our light meter here, 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 each time counting the number of organisms, for example daisies or whatever we were looking at, and calculating the light intensity. Now I want you to imagine that we're sampling in a woodland, and along the transect away from the tree there's lots of long plants and long grasses. Now in this case we simply can't place a quadrat on top of the long grass, okay, because the grass is just far too long. So you might see in the exam them talking about counting the plants that are just touching the transect line. So if you've used string and pulled that along the long grasses, you might be simply counting the different species that are touching the transect line. Another thing to look out for, don't be put off if they talk about wanting to measure something vertically. So for example, they could look at how height affects leaf size or how height affects lichen coverage. In this case, think about doing the practical in exactly the same way. So you may use a very small quadrat and measure percentage cover of something as you go up, or you might simply, if you're measuring leaf size for example, see any leaves that were touching the line and measure their size as you go up and see how that changes as you go up the transect. So don't be put off if they're talking about one vertically going up a tree or up a wall or something like that. It's exactly the same process as the horizontal transect that we looked at before.
In paper 2, you need to remind yourselves of the topics of homeostasis, which is topic 5, inheritance, variation and evolution, which is topic 6, and ecology, which is topic 7. So let's look at the main parts of all those 3. So homeostasis and response, first of all. Homeostasis means maintaining a constant internal environment. This is important to maintain the optimal, which is the best conditions for enzymes. So enzymes working in your body, for example, work best at 37 degrees C. So we'd want to maintain that temperature at exactly 37 degrees C. Not only do we want to keep body temperature the same, but we'll also want to keep things such as our blood glucose, so our blood sugar levels, and our water levels within our blood and within our body cells at constant levels. In your exam, a common question is asking you to compare nerve and hormone responses, because to maintain a constant internal environment, you might be looking at a nervous response to do this, or you might be looking at a hormonal response. So I want you to concentrate on the types of words that you need to use in a compare answer. So let's have a look at this first line here. Nerve responses are faster than hormone responses. If you're asked to compare, you must use comparative words. You can't just say nerve responses are fast. You have to make sure you're comparing it to hormones. Hormone responses are longer lasting than nerve responses. Nerves act on a precise area, whereas hormones act in a more general way. If you can't use a comparative word, use the word whereas to link your sentence. And this one, hormone responses are chemical signals, whereas nerves are electrical signals. So a common question asking you to compare nerve and hormone responses. Let's delve into a little bit more detail about nerve responses now. So it starts off with a stimulus. Now that is any change in the environment. It could be a chemical stimulus, it could be light, it could be sound, or pain, for example. Something to do with your senses. The first stage in the nerve response is the receptor. So you have receptor cells in your body that detect a change in the environment. And these are sent along the first neuron, which is the sensory neuron. So think to yourself, if you're sensing something, you're using the sensory neuron first. This goes to your central nervous system that coordinates the response. And then it goes to your motor neuron that is going to do something at the end of the pathway. And that motor neuron sends a signal to an effector, and we'll talk about what those can be in a minute. and that brings about some kind of response. So that's the general pathway that you need to remember. Let's say, for example, that someone was throwing something at you. The stimulus for that would be light reflecting off of the object. You'd detect that by light receptor cells in your eye. You'd send the message along the sensory neuron to the central nervous system, which comprises of the brain and the spinal cord. And then you'll go to a motor neuron. An effector in this case would be muscle, and your muscle would contract and move, and that might either to be catch the ball or move out the way, whichever it is. Now in this case, I decided to concentrate on an effector being a muscle. There's two things that an effector can be. It can be a muscle or it can be a gland. So interesting link between the nerve and the hormone pathway, in that actually a nervous response can result in a gland being used to release a hormone. And where this comes into play is normally the adrenal gland that will release adrenaline in a very, very quick response, okay, a fight or flight response. Now, when those neurons are sending the signal along, they might have to pass the message from one neuron to another. And when they do that, they have to pass the signal across a synapse. A synapse is a gap between two neurons. So this here is the synapse. And the message within the nerve is a very quick electrical message. So either side here, the message will be transported in an electrical way. And in between, there is a chemical message. Okay, so as the nerve is carrying the signal along here, it's electrical, it gets to the end, chemicals are transmitted, which slows the whole process down slightly, and then it gets transferred onto the next neuron. So the more synapses that you have, the slower the response will be. And in the exam, they might talk about using the equation speed equals distance over time in relation to how quick nerve responses are, but they'll just expect you to know that anyway from Key Stage 3. So reflex response then, sometimes you have a very, very quick response and it doesn't go to the brain to be coordinated. So normally our central nervous system is means that the signals go up the spinal cord to the brain and that coordinates the response. That's the coordinator. However, in this case, the message just nips across the relay neuron instead, which is in the spinal cord. So you might see a diagram that looks a little bit complicated like this. All this is showing is this part of the spinal cord. And here we have our stimulus here. We're getting stung by the bee, and our pain receptors are detecting that, sending the message along the first neuron, which is a sensory neuron. Here we've got a synapse, and in the middle, this neuron here is the one called the relay neuron. You might be asked to annotate a diagram like this in the exam and label the neurons. So rather than going up to the brain, it doesn't do that. It whips across the relay neuron and goes straight to the motor neuron. And again, the effector might be a muscle or a gland. In this case, a muscle which will contract. So moving on to the hormones then. Hormone responses. Hormones are secreted by glands in the endocrine system. Okay, so the endocrine system is a system of glands. The most important gland is the pituitary gland, because we call that the master gland, because that can switch on and off other glands by releasing hormones. So this one's the master gland. Hormones that are released like this include hormones such as LH and FSH. The thyroid gland is in your neck. That produces thyroxine, which controls your metabolism. The adrenal glands are just above the kidneys there. That releases adrenaline. The pancreas releases many hormones, and make sure you're talking about hormones and not enzymes when you're talking about the pancreas in this paper. So hormones such as insulin, for example. The ovaries produce estrogen and progesterone, and the testes in males produce testosterone. So you might be asked to label a diagram or say what is released by each of these glands, remembering that it is the endocrine system. So as we said, the response in the hormones is a lot slower. One thing that they can do is be involved in controlling blood glucose. And for that, it's the pancreas here that's the important gland for controlling blood glucose. And we said that was an important thing to control for homeostasis. So if your blood glucose is too high, insulin is released by the pancreas. Okay, so insulin is the hormone. It's released by the pancreas into the blood. All hormones are chemical messages or chemical messengers. They're carrying the message. So chemical messengers that travel in the blood. So they're dissolved in the blood plasma, and that's how they travel around. When insulin is released by the pancreas, that causes excess glucose in your blood to move from the blood into your liver and your muscle cells. So it takes it out of the blood for you. And there it is stored as glycogen, because glucose is soluble. So you can't store anything just as glucose. You have to convert it into a larger molecule like glycogen, which happens in the liver and the muscle cells. So for foundation, that's all you need to know on the blood glucose. For higher tier, I'll go into it in a little bit more detail later on. So linking into this, we've got two different types of diabetes, which is all concerned around this hormone, insulin. So with type 1 diabetes, that's the one that you inherit. The pancreas produces little or no insulin. So you may well have type 1 diabetes yourself, or know the people that are affected by type 1 diabetes. And you may well know that the treatment for that is with insulin injections or an insulin pump. So they can't produce it, so they need to inject that into their bodies to stop their blood glucose levels getting dangerously high. Type 2 diabetes then. If a person becomes resistant to their own insulin, that is when they are developing type 2 diabetes. So the doctor in this case won't give somebody insulin injections. That's not how they'll suggest that for the diabetes to be treated, because it's all to do with their lifestyle and what they're doing to have caused this type 2 diabetes. So what they'll suggest the patient should do is control the amount of carbohydrates that are going into their diet and make sure that they exercise more. And in that way, they can control their own blood glucose levels. So hopefully it won't have any other effects on their body. Okay, moving on then. Menstrual cycle. So with these menstrual cycle, the main hormones that we're talking about are FSH, and that causes the egg to mature, and LH, that causes the egg to be released. So these two hormones, I want you to remember the abbreviated ones, the FSH, the LH, they are produced in the pituitary gland. And then we've got two other hormones. We've got estrogen, which stimulates the lining of the uterus to start growing, and progesterone, that maintains the lining of the uterus. So these two are produced in the ovaries. So if you want a little visual here, think that the abbreviated words here at the top, that's the pituitary gland, and the other two longer words here down in the ovaries. So what happens? The estrogen causes the uterus lining to build up, and the progesterone helps to make sure the uterus lining is maintained before the next menstruation, when the uterus lining breaks down. On day 14, that's when there's high LH levels, because that causes the egg to be released at day 14. And before that, the follicle stimulating hormone travels down in the blood to the ovaries and stimulates an egg to mature. And obviously the woman would just want one egg per month to mature. I'll go in more detail about how these interact with each other for higher tier later on. Controlling fertility then, still using hormones. There's hormonal methods that you can use. And the hormones that we want would be estrogen, because they inhibit, which means stop, FSH. So if you haven't got any FSH, you can't mature an egg. So you're not going to get pregnant. Progesterone is the other hormone that you can use. That inhibits FSH. So the egg, again, can't mature and be released. So both of those are hormones that can be used. Now, the progesterone also causes a sticky mucus just in front of the cervix to prevent any sperm from reaching the egg. So the progesterone does two things. Now, you could put those hormones, you could put one or the other or both of them into a pill, a patch, an implant, or injection. So these are our hormonal examples of contraception. You could use an intrauterine device, which is otherwise known as a coil as well. And there's this one is one that will be inserted into the woman's uterus and it will release hormones. Non-hormonal methods then, you've got barrier methods such as condoms, female condoms, and diaphragms. And these are otherwise known as barrier methods because they physically block the semen from going into the uterus. So you might see those as barrier methods. And alongside those, you can use spermicide to make sure you're getting the best contraception possible. And spermicide is just a chemical which kills the cells, the sperm cells. Another method you can use is copper coils, essentially copper intrauterine devices. So they actually kill the sperm rather than releasing hormones. So they kill the sperm instead. And there are methods such as permanent methods such as sterilization that you could use for the male or the female. Abstaining, which is avoiding intercourse or avoiding intercourse when you're going to be most fertile. So avoiding intercourse around that day 14 when the egg's released will help. Okay, meiosis then. Meiosis is paper 2. You would have seen mitosis in paper 1. This is the one word at GCSE where they expect you to spell it correctly, because if you spell it wrong, you're not telling the examiner that you know whether you're talking about meiosis or mitosis. So if you remember in mitosis, that was for growth and repair. So everything from the embryo onwards to growing an individual is mitosis. However, meiosis has a very specific role, and that is to produce the sperm and the egg, so to produce the gametes. Now, in the exam, they might not well talk about humans. Okay, in humans, we have 46 chromosomes, and after meiosis, we produce either an egg with 23 chromosomes, if we're talking about a woman, or a sperm with 23 chromosomes. Okay, so we're halving the number of chromosomes that we've got, so that when they come together in fertilization, you make 46 again. But just for you to be resilient, they might not ask you about humans in the exam. They might ask you about a completely random animal, or they might even start talking about plants or something else. They'll probably keep it on animals, to be honest. But here we go. A fruit fly has 8 chromosomes. How many chromosomes would you find in a fruit fly egg? Well, it's exactly the same principle. If he's got 8 to start off with, in the egg, there'll be 4 because there's half the number of chromosomes in the gametes. So let's start and look at what meiosis is with this example of a fruit fly, because it's easier to draw 8 chromosomes than it is to draw 46. So let's look at this fruit fly. It's got 8 chromosomes. Meiosis happens in either the ovaries, so meiosis happens in my ovaries, will help you remember it, or my testes if I'm a boy. So ovary or testes. So we start off with 8 chromosomes in a body cell within the ovary or testes. Those chromosomes, just like mitosis, will have to double up. But the first thing that they do is they line up in pairs. So we've got pair 1, pair 2, pair 3, pair 4. So if this is a human, the pairs would go down to 23. And at this point, they start to switch over their DNA from chromosome to chromosome. So there's some switching, which means the chromosomes will look a little bit like this. And this switching here is what causes variation. It's what causes all of the eggs to be different and all the sperm to be different genetically. So the first division that they do is pulling these chromosomes apart after they've mixed up their DNA like so. So that's the first division, division 1. The chromosome pairs pull apart. You can see they're all slightly different now. Then we have a second division whereby these chromosomes pull apart, pull to the edges of the cell like so, and you end up with 4 gametes at the end of it. So you can see here we started with 8 at the top and now we've got 4 at the bottom. They are all different, okay, so they contain variation here. They're not the same chromosomes in each one, so they're not identical to each other. And this is the main summary of meiosis. They're to produce the gametes, which are the sex cells, which in animals are the sperm and egg. They take place in the ovaries and testes, and it causes variation in the offspring. So here we've produced 4 cells at the bottom. Sometimes we call these daughter cells. So 4 cells at the bottom. We started off with 1 cell, and there is 1, 2 cell divisions. Okay, just count these as stages. 1 division there, and a second division there. Reproduction. We'll start off with fertilization first of all. Fertilization is the fusing of the nuclei of the sperm and egg. Sexual reproduction then is what involves fertilization. So when a sperm, for example in a human with its 23 chromosomes, fuses with the nuclei of an egg with 23 chromosomes, they will make a fertilized cell with 46 chromosomes. That will then start to divide by mitosis to make an embryo. So mitosis when it's growing into an embryo. Meiosis when it's to make these first of all. Sexual reproduction then, the offspring are genetically different because you've got a genetically different sperm to its father and a genetically different egg to its mother. When they combine, you're going to make different offspring. So they produce variation. And then you've got asexual reproduction, where the offspring are genetically identical to their parents, okay? They are clones of their parents. Some animals can do both asexual and sexual reproduction, and that's the same for things like plants and fungi as well. A lot of animals, however, can only do the sexual reproduction. Just to recap, in animals, the gametes, the sex cells, are the sperm and the egg. But in case they ask you in the exam, they also want you to be aware about plants, in whereby the gametes are pollen and egg. So don't talk about sperm when you're talking about plants. DNA then, moving in further into chapter 6, we need to know about DNA. So first of all, if we remember what a cell looks like, the cell has a nucleus, and that is where the DNA is kept. And the majority of the time, the DNA is loose within inside the nucleus. And at certain times, like cell division, it will make chromosomes and wrap itself up into little packages. So the DNA stands for deoxyribonucleic acid. That's the chemical that makes up DNA. It's a polymer, which means it's a long chain. And it's a double helix. So it will look something a little bit like that, where you've got one strand and then a second strand wrapped around it like so. So that's what our DNA looks like in its structure. A genome is something that we need to know about as well. You might have heard of the Genome Project, whereby they've mapped all of the genes in the human body. So the genome is the entire set of genetic material in an organism. So it could be a plant, an animal, or a fungus. If you're talking about a genome, it's all of the genetic material, so all of the DNA within an organism. So our DNA within our nucleus codes for all of our genes, okay? And these control all of our characteristics. A gene itself is just a length of DNA. So if that was our DNA, a gene would be a section here of DNA, and that codes for different characteristics like hair colour or eye colour and things like that. The gene is used to make an amino acid. And if you think of the opposite of the digestion module, amino acids can make proteins, because in the first paper we talked about proteins breaking down into amino acids in digestion. So you need to think about it the other way this time. Genes code for amino acids and those make proteins. So the amino acids are the short units and they would join together to make a protein like an enzyme or a hormone with a specific shape and active site, like you would have looked at in paper 1 where we talked about proteins having a specific active site. So moving on with this chapter about DNA, we're going to look at inheritance. And first of all, we'll look at inheritance in terms of whether you're going to be a male or a female. And that's all to do with your X and Y chromosomes. And in humans, that is chromosome pair 23. So we've got 23 pairs, and the last pair determines our sex, whether we are male or female. So females have 2 X chromosomes, and males have an X and a Y chromosome. So when a female and a male reproduces, we can work out the chance of them having a male or a female. So you might be asked to complete a genetic cross like this using the sex chromosomes. So we've got the XX here, we've got XX here, and then we've got XY and XY. So we have got 50% chance of being a male and 50% chance of being a female. So just write that somewhere near the diagram to finish off the answer. 50% male and 50% female. You may well see crosses in diagrams like this as well. This tends to be called a Punnett square, this one. And you might see diagrams a little bit like this as well, whereby they're splitting up and you're having your XX here. Let's go back. You'd have your X and your X coming together, your X and your Y, your X and your Y, and your X and your X. So still the same chance, obviously 50% chance of male, 50% chance of female, but you may well see the diagrams represented like that instead. Genetic crosses then. They could talk about inheriting absolutely any characteristic when they talk to you about questions about genetic crosses. So I've just made up something random. Think about mice and think about an allele, which is a type of gene, or a form of gene, we should say. So allele is one form of a gene. That's the definition that they'd want you to write, one form of a gene. So let's say we had one form of a gene that could give you thick fur, and another form of the gene which would just give you normal thickness fur. So in this case, I've done a capital T for thick fur because this is the dominant allele. So if this is present, the animal will have thick fur, and this is recessive allele. If it's recessive, this means that if it's just present on its own, it won't have normal fur. It has to have both alleles present, like TET, lowercase, lowercase, in order for the animal to have normal fur. So let's look at some genotypes versus phenotypes. Genotypes are your genes, and phenotypes is what characteristic is expressed. So this means it's going to be a word to describe it. In this case, our characteristics are thick fur or normal. In your exam, you could be talking about blue eyes or brown eyes. You could be talking about red hair or black hair. That's your phenotype. So let's have a look for this one. Homozygous dominant. That means homo is the same, okay? So 2 of the same dominant. That would give a phenotype of thick fur in this case, because that's the dominant allele. Got homozygous recessive, which means 2 of the same, homo recessive, so 2 lowercase letters. And this is the only opportunity for you to get a normal fur mouse. And the final combination you could have is heterozygous. So that means 1 of each. Hetero, 1 of each. So you get a capital T and a lowercase t. But You need to look at which one's dominant. Dominant is thick fur. So wherever you have an allele with a capital, that means that you will express that characteristic. So in this case, because we've got a capital, that wins over, so this heterozygous will be thick fur. You could be asked to complete any genetic crosses. So for foundation, they will give you the alleles. So they could give you homozygous dominant, for example, homozygous recessive, and you would just need to complete the cross by matching up here. So we got TT, if we look across at these two, TT, TT, and then TT. So in this case, we've got 4 heterozygous, so we are going to have 100% thick fur. If we do another example where we've got heterozygous, heterozygous, when we cross those, like so, we will have 25% chance of it having normal fur. So you need to complete the cross and then write down the percentage chance for the characteristic below. If in doubt, write both of them, but in the exam they'll kind of tell you which characteristic you're talking about, whether they want you to write about thick fur or being normal. Polydactyly and cystic fibrosis are 2 disorders that you need to know about in this genetics topic. Polydactyly is a dominant allele. So if somebody has the allele for polydactyly, they will have this disorder. And this disorder means that somebody has extra fingers or toes. So this will happen to a few people at birth, and they'll just get removed straight at birth, their little extra digit that they've got. And yes, it is dominant, but this disorder is very rare. So if you've got the gene in your family, you'll be likely to pass it down, but it's very rare allele. So not many of the population have this allele. Cystic fibrosis, however, is a recessive disorder. That means you could have 2 parents that carried the allele. So let's just do capital B for not cystic fibrosis and little b for having cystic fibrosis, like so. If those 2 parents were going to have a baby, they themselves would not have cystic fibrosis because they've got a dominant allele for not having the disorder. However, if those 2 people reproduce and had a baby, because of the genetic cross, which we did a heterozygous one before, you would end up with 25% chance of their child having cystic fibrosis. So sometimes 2 parents have a child and they have no idea that they are carriers of this recessive allele. It means they're carrying it, but they're not expressing cystic fibrosis. So it's a recessive one. It affects cell membranes, so it disrupts the function of cell membranes. So it causes sticky mucus to build up in air passages and the pancreas as well. Now, with these genetic disorders, there's some recent technology that has suggested we might be able to do some embryo screening. So an embryo is just a small ball of cells right at the start of life. So just after the sperm has met the egg and made a fertilized cell, that will divide by mitosis and produce an embryo. And the idea is that parents could have the option, if they know that there's a history of a genetic disorder in their family, they could have the option where they produce a child by fusing a sperm and the egg together. But at the embryo stage, a doctor could remove some of the cells and screen them, which means check them for genetic disorders. And then the idea would be that they would pick an embryo that doesn't have a particular genetic disorder in. So there's lots of moral implications with this. Some people say that you shouldn't be able to choose what child you have, but obviously there's positives with you bearing a healthy child, and later down the line, even though it's expensive, it might reduce the costs on the NHS for further treatments if somebody did have the disorder. On to the evolution topic. With every evolution question, what I would like you to do is almost ignore what animal they've got in front of you or what plant they've got in front of you. don't be put off if they start talking about axolotls or limpets or lichen or things that you've never heard of before. Think about the framework for evolution by natural selection and try and get these answers in no matter what. So the idea that there's natural variation in the population anyway, and a mutation, which is a change in the DNA, causes a change in phenotype. If you remember, that's what the animal looks like. So if you can't remember phenotype, it's what the animal or plant looks like. So individuals best adapted to the environment will then survive. So get the words best adapted and survive in there. They will reproduce and they will pass on their genes to the next generation. So absolutely any question, regardless of the animal or the plant that they're talking about, if they're talking about evolution by natural selection, you need to get those steps in. Now, when they're talking about a mutation causing a change in what the animal looks like, then you can link it to the question. So, for example, if a mutation caused a snake to be green, that might make it camouflage in the grass better. If a mutation caused a marine iguana to be black, again, that would cause it to camouflage better into the rocks. Or it could be a mutation that caused something to have wings and allowed it to fly, or whatever it is. So the last thing to do is relate it to the question itself. Speciation then, leading on from natural selection. If you have isolation, so two populations, let's take some sheep, for example. Just by nature, some of those sheep will be naturally fluffier than the other ones. So let's do some normal sheep and some really fluffy sheep, like so. If those sheep were to be separated, they could be separated by a mountain range or a river or a lake, for example. That suggests a river, large river, became between two sheep populations. We might have one on this side, like so, and one on the other side. Now, at the moment, these sheep can still interbreed with each other and make offspring. But because of the isolation, because they can't get across the river, these might end up on two different islands and they might gradually drift away from each other. When they do, they might find themselves in slightly different climates. It could be that there's different food on the island for them to eat, and they might adapt and evolve to eat those foods. Or it could be that there's different environmental differences. For example, on this island, let's say that they drifted off to somewhere that was very sunny. If you think to yourself about evolution by natural selection, think to yourself which sheep would survive best on this island. Well, probably this sheep here is the one that is best adapted. So that will survive, reproduce, pass on its genes to the next generation. So gradually over thousands of years, these thick-furred sheep would die out and you'd have the lesser, the normal sheep or the non-thick-furred sheep surviving and reproducing and being more common in the population. So you could get a change where actually when these sheep were to meet again, it could be that they can't breed anymore because they're so different that they're actually now a new species. So if they're talking about making a new species, talk about natural selection that we talked about previously, but add in the word isolation. And if you end up making a new species, by definition, if two individuals are classed as different species, if they can't reproduce and produce fertile offspring, that's a key definition for what makes a different species. So if these came back together a few thousand years later and they were very, very different looking, but they could reproduce and make fertile offspring, then they would still be the same species. But if they came together and they changed so much that they couldn't reproduce, they might be able to make babies, but those babies can't make their own babies, so they're actually not fertile. Then we would call them two completely different species. Selective breeding then, a couple more things. So we'll talk about selective breeding. That's when you are choosing the animals that you want to breed together or choosing the plants that you want to breed together. So you're playing with nature, you're playing with the natural selection cycle, and you're not letting nature choose those animals that are best adapted. You are choosing them for various reasons yourself. So you'd select a male and a female with desired characteristics. But for example, if you wanted a cow to produce lots of milk, you'd need to talk about not selecting a male that would produce lots of milk, but with this case, a male whose daughters would produce lots of milk. So in this specific example, you'd either choose the male and the female with a desired characteristic, or where appropriate, whose offspring have a desired characteristic. Okay, so if you're talking about cows and milk, obviously a male's not going to produce milk. So you'd choose a male that has daughters that produce lots of milk and breed that with a female. You breed these individuals together, and from the offspring, you select those individuals with your desired characteristic. It could be producing more milk or more meat or whatever it is. And then you breed those. And then you continue this process for many, many generations. So always picking your individuals that you want to breed and breeding them together. So you might want to do this for a higher milk yield. So yield is how much you get. So getting more milk from cows, and that's what we've done over several years and several generations of cows. You might want to get a higher meat yield. So we've done that with things like chickens and continually bred them until they're producing us more meat. Or in the plant world, maybe something that's got bigger flowers or better flowers instead. If you don't want to do selective breeding, this is something we've done for hundreds of years, but more recently, technology has allowed us to do something called genetic engineering. And with genetic engineering, you take a gene from one organism and insert it into another. Now, you can really let your imagination go wild with this. So there are stories of people taking a fluorescent protein out of jellyfish, for example, and inserting it into cats to make glow-in-the-dark cats. You can literally take any gene from one animal or plant and insert it into the genome of another one. Now, you need to do that at a very early stage, like when it's an embryo. You can't just stick it into an adult animal and expect it to change. You might want to do this for crops so that they're resistant to herbicides, for example. Take a herbicide-resistant gene and put it into your wheat crop. Crops that are resistant to disease. Perhaps crops with increased yield. You can tell that I'm talking all about plants because the mad example of the fluorescent protein in a cat is not really a moral one. So we tend to do this on plants rather than animals. And bacteria, for example, to produce insulin, you can take the insulin gene out of a human and put it into bacteria. And I'll go into this in more detail for higher tier later on. These, when they are genetically engineered, are called GM crops. Okay, genetically modified crops are those that have been genetically engineered. So classification. Here's a little bit of a rude rhyme to help you remember classification. So king penguins crap on French girls' skirts. Okay, so that mnemonic there will help you remember how we classify animals. So, and plants and fungi and everything else. King stands for kingdom, P for phylum, C for class, order, family, genus, species. So this classification system here is called the Linnaean classification system, and it was developed by somebody called Carl Linnaeus. So he came up with this system, and all of the animals and plants and fungi and bacteria that he could see, he put them in this system. However, another Carl came, okay, it's one for the Carls this, another Carl came, and he said, I think there should be something above kingdom. So a guy called Carl Woese, this time, W-O-E-S-E, came up with the three-domain system. So he said, above kingdom, we should have three domains called eukaryota, where all the eukaryotes are, so plants and animals. All the bacteria should be in a separate one. And this is your true bacteria, what you know as those kind of single-celled things. And then archaea is the third domain. Now, these two are very similar because they are actually both bacteria. But this one you call the true bacteria, and this one is very primitive bacteria. These are where your extremophiles are. Extremophiles are bacteria that live in extreme environments like high pressure, high temperature, high salinity. So he added these domains. And then under eukaryota, you still have the kingdom, phylum, class, etc. But this is now on top. Now, key is they like questions about why you change the model and why you change the system. And it's all to do with new evidence coming to light. Okay, so if they ask you about why the system's changed, talk about new evidence. So the advances in microscopes and things like that let us look at the subcellular parts of animals and allowed us to compare whether they had nuclei, whether they had ribosomes and mitochondria and things like that, which then allowed us to separate them into other groups. And it may well change in the future. Right, last thing on this then is the binomial name. The binomial name, bi for two, like bicycle, nomial name, comprises of the genus and the species. So, for example, for humans, our genus is Homo and the species Sapiens. So that would be our binomial name. Take a barn owl, for example. You don't need to remember these examples, but if they give you the binomial name as a barn owl of Tyto alba, they might say to you, what's the genus? So the first word is the genus, so you'd write Tyto, and for reference, that word is the species. So those two together give something its binomial name. So they may well give you a table and then say, write the binomial name. So you just take the genus and the species from the table and write those together. A few more bits on ecology then. Ecology topic, it starts with a lot of words. So let's start off with population. Population means that all the organisms of a single species living in a habitat. So if we talked about fish, for example, this would be a population of fish. A community is populations of different species. So you might have a fish in there, you might have a different species of fish, a much larger predatory fish, for example, and maybe some plants in there. That would be a community. An ecosystem is the interaction of living, biotic, and non-living, abiotic factors. So it could be that you're talking about the rocks that are in that system, the temperature of the water, the current of the water, and adding all the abiotic things in there as well. And two more keywords, biodiversity. Bio means living, diversity means variety. So biodiversity is the variety of living organisms on the earth. Final word, interdependence. That is the dependence of different species on each other. So some species could be dependent on each other for food, perhaps for shelter. So birds are dependent on trees for shelter, for example. Pollination. So bees depending on particular species of flowers depending on particular species of bees for pollination, for example. So we talked about abiotic and biotic factors briefly. Biotic factors are the living ones. Biology is the studying of living things. So bio means a living factor. Predators are an example of a living factor. Pathogens, so causing diseases, competition between each other animals, and food availability. So if you think about these donkeys here, they would be competing with each other perhaps for food, and this would affect their survival. Abiotic factors, non-living factors, lots to do with weather on this one. So temperature would affect that ecosystem, wind intensity, amount of rainfall, pH, salinity, which is how salty it is, and oxygen availability, for example. Have a look at what situation you're given, because if you're talking about oxygen availability, you're only talking about aquatic environments. So if we're talking about the water here, we'd be talking about things like how salty it is and oxygen and temperature. but obviously we wouldn't be talking about wind with that one so much. Adaptations then, they could ask you again about adaptations of any animal or plant. So just look carefully and use your knowledge of what's in front of you to think about things that are behavioural, structural, and functional. Behavioural is how the animal behaves. Structure is what it looks like, what its structure's like. And functional is anything that happens inside the body. So anything to do with its metabolism, or whether it respires, or the processes that happen inside the body. So for example, if we take this bat here, we could talk about behavioural. It hunts at night to catch its prey. It roosts in groups, for example, for insulation. You don't need to get this specific example down, but I'm just showing you how you could answer this type of question. Structural, that's what it looks like. So large wings for flying, thick fur for insulation, brown fur for camouflage. And functional might be something like the fact that it hibernates in winter. Internally, it lowers its body temperature. So whatever animal or plant you're given, try and think about, could be a 6 marker, for example, try and think about how it behaves, the structure of it, and things that happen inside of it. Water cycle then, nice and easy. It just needs to it needs for you to recall 4 words. The first word is evaporation. So from seas and lakes and things like that, the water will evaporate. It will then move up into the sky, where it will condense together to form clouds. And then at a certain time, you will get the rainfall, which we call precipitation. So don't just talk about raining, use this posh word here, precipitation, instead. And the final factor is to do with these trees. From paper 1, you'll know that trees and plants also lose water, and that process is called transpiration. So 4 things to consider about the water cycle. We also cycle carbon. Now, carbon is a chemical that is in an element, that is in lots of plants and animals, the majority of plants and animals. So we just need to talk about here about the way this is cycled around our planet. So one way is feeding. So if this cow here eats the grass, then the carbon is being transferred from the grass into the cow. Photosynthesis. So if we think about carbon being in carbon dioxide, then the plants will take in carbon dioxide from the air, and that's part of the carbon cycle, and they'll obviously release oxygen. They'll produce glucose in that process. Glucose has carbon in it, C6H12O6. So when the animal eats the plant, then they'll be taking that carbon up as well. Respiration, both animals and plants respire. And when they do that, they will be releasing CO2 to the atmosphere. Combustion, if we've got fuels inside the tractor here, for example, in factories, cars, if we're burning fossil fuels, we're releasing CO2 into the atmosphere. And decay. Now, this is something I do want to highlight, because this is a common theme in the exam, talking about decay. Decay is caused by microorganisms, so small organisms, things like bacteria, etc. Now, microorganisms are involved in this cycle as well, because they also respire. Just because they're really small, don't count them out. They respire and release carbon dioxide. So when the dead plants and the dead animals are decaying in the soil, the microorganisms are contributing because they're respiring when they decay them, and they release CO2. In global warming, I want you to remember, from the sun, short wave radiation is released. This comes up in the physics topic as well. Short wave radiation is released. That goes through our blanket of greenhouse gases in our atmosphere, but it is reflected back after it's reabsorbed and then re-emitted from objects on the Earth as long wave radiation. But the trouble is, the long wave radiation finds it very difficult to get through this layer of greenhouse gases. So what it does is it's reflected back down to the Earth, and that's what causes it to warm up. And the consequences of that you should be aware of from the news, things like flooding and extreme weather and hurricanes and things like that. Waste can come from a number of different ways, and it's mainly caused by the fact that our human population is rising at a very, very fast rate. So it's like that for thousand years, and then suddenly we are increasing at a very fast rate. We're producing toxic chemicals, solid particles in the smoke that we release, sulfur dioxide in the smoke that can cause acid rain, nuclear waste, and sewage. And we're having trouble at the moment trying to get rid of these and store these so that we're not polluting and damaging the planet. Now, one of the ways that I want to dwell on, because it does come up sometimes, even though it's not made obvious in your spec, is eutrophication. So you can see here we've got a really green lake, and that green lake is covered in algae. So what happens is fertilizer can run off from the farmer's land. So lots of nutrients in the soil here going into the lake or the river. The plants and algae love those nutrients, so they use the nutrients from the fertilizer and they bloom and they multiply and they cover the lake. When they cover the lake, this reduces the sunlight that can get in. And this means that the plants in the water die because they can't do photosynthesis anymore, so they die off. Again, relating to microorganisms now, the microorganisms that are in the lake decay the dead plants. And the link that I made before, these microorganisms respire. And when they do that, they use up the oxygen in the lake, and this can cause lots of larger animals to die. Like the larger fish and things will now die because the oxygen levels get so low.